Good afternoon to those of you tuning in from the UK and good morning to those of you in the US. We are broadcasting live today to talk about metabolomics and in particular precision metabolomics. If you've been out to any of the big events this year, you'll probably have noticed Metabolon making a big impression on exhibition show floors all over the place. But it's not just a great marketing campaign. Metabolon have had a very busy year helping labs fit in metabolomic analysis into their workflows to great effect. And that's what we're talking about in this webinar, using controlled processes to provide higher accurate data to map metabolic changes that can help diagnose and map treatment with greater confidence. Taking you through how this works, we are delighted to have with us Metabolon's Vice President of Laboratory Operations, Luke Miller. Luke joined Metabolon back in 2013 after 17 successful years at GlaxoSmithKline, where he led the Analytical Chemistry Department. Throughout his career, he has experienced a number of highs, including the development of supercritical fluid instrumentation and technology for analysis and purification of small molecule drug-like compounds. In addition, since his arrival at Metabolon, he has isolated a novel biomarker with potential for diagnosing pre-diabetes. Not only that, he has also led the team responsible for fully categorizing it. So I imagine it's pretty reassuring to hear that you're in really great hands today. On that note, it's over to you, Luke.
Thank you very much. Good morning, everybody, and thank you for joining me today on our presentation of the Metabolon Precision Metabolomics platform. Today I'm going to be discussing the processes that we've put in place in Metabolon to allow us to analyze N of 1 or single patient samples on our untargeted metabolomics platform. The talk will be in 3 parts. I'll discuss the processes and controls that we've put in place to bring the median RSD from our platform to less than 5%, while increasing the coverage of small molecule metabolites analyzed via that platform. I'll then demonstrate the power of the platform with some examples and case studies, and then finally I'll discuss what Metabolon is doing next to enhance the data that we obtain. I'd like to start with a quick overview of the company. Metabolon was formed in 2000, so we've been around for 17 years. We are headquartered in North Carolina. We've recently branched out to place platforms at sites around the world, including in China with Dian Diagnostics, in the Middle East with the anti-doping lab in Qatar, and in Germany. We're almost 200 employees worldwide with experts in biochemistry, mass spectrometry, and the software that we've developed. The Metabolon Precision Metabolomics platform is much more than just a set of mass spectrometers analyzing different matrices. At the core of our untargeted precision metabolomics analysis is a set of processes and controls, software, hardware, and infrastructure that have been carefully designed to reduce the errors from the analysis and produce the best quality metabolomics data in the industry. This includes controls in all aspects of the process: sample prep, data acquisition, data processing, and interpretation. The first part of our process is not really sample prep, although I've highlighted that on this slide. In fact, our study managers are involved in the design of experiments before samples are collected and even sent to us. Once the design is complete and the experiment is underway, we work with our clients to advise on sample collection and shipping to ensure consistency across all samples. Once samples arrived, they are inventoried and logged into our internal LIMS system, tracking everything from where they're placed in the freezer and then as they move through our processes. The samples go through a very standardized extraction process, which includes the addition of multiple internal standards to control both the extraction and the analytical processes. Analysis takes place on 3 Q-exactive LC-MS systems running 4 analytical methods, 2 in positive and 2 in negative electrospray mode. Each method is designed to both capture unique metabolites and provide redundancy, so approximately 80% of the metabolites analyzed are seen on more than one method, allowing another quality control step in our process. The 2 positive electrospray methods, each running for 3.5 minutes on 1 Q-exactive, analyze respectively for polar compounds and lipid compounds. The first negative electrospray method is a 7-minute reversed phase UPLC method, capturing the majority of those metabolites that ionize under negative conditions. The second negative method is a 7-minute Hillick method designed to capture polar metabolites such as small organic acids and sugars. It produces separation of sugars and sugar phosphates, something that's not usually capable within the industry. Our runtime, therefore, is 7 minutes per sample, allowing us to carry out 180 injections per day, 140 of which are client samples and 40 are QC samples. This gives us a total capacity with 6 platforms now in operation of over 200,000 samples per year at our RTP headquarters. And obviously, we've got 35,000 sample capacity in China, the same in Qatar, and the same in Germany. All of our instrumentation is maintained in a ClearCap certified laboratory in a totally controlled environment. We control the temperature in the lab to 68 degrees Fahrenheit, plus or 2 Fahrenheit. The voltage to each instrument is smoothed to plus or minus 1 volt. Goes through a UPS system and a backup generator. So we have 24 hours of backup power to each one of the mass spectrometers, and then a natural gas, mains natural gas generator that goes to all of our fridges and freezers, allowing us indefinite power supply to the fridges and freezers. I like to joke to anybody that comes to see us here in North Carolina that the lab will keep operation 24/7. You won't be able to get a cup of coffee if the power goes out, but the lab will keep working. Within 76 seconds of the data being collected, data is removed from the instruments, archived, and run through our library searching software. One of the most important parts of that, then, is quality control of the sample data. What you're seeing here is water blanks that are run every set of samples that we run. In fact, we run 6 QC samples with every sample set that we run. Here you can see the importance of doing that. What you're seeing here is that some of the major peaks that we analyze are in fact coming from the water that we utilize in the analysis, and the most important real biological signal are those 2 small peaks that I've highlighted on the slide. As I mentioned, we run 40 QC samples, including water samples, every set of samples that we run every day on every instrument. We've written our own peak integration code to better provide peak integration and detection following our analysis. One of the things we've come to understand is important, not necessarily just to be able to integrate every peak, but to get consistency of integration. What you're seeing here in this example is the analysis of a 1 and 2 form of a particular phosphatidylcholine that we've analyzed. As the 2 metabolites are being impacted in the study that we're showing here, what you're seeing is that the peak itself changes for each of the 2 metabolites. The importance of consistent integration then is that these changes are measured consistently. Here I'm showing 9 of the individual integrations of these 2 peaks, and what you can see is that 3 of the integrations are incorrect. In each case, the 1 form has been completely lost, leading to incorrect integration and measurement of the 1 form, and either up-regulation or down-regulation of both the 1 and 2 forms. Our software allows this to be corrected, making sure that what happens is consistent for all peaks. Now, I make note that this isn't necessarily the best integration that we can get for each one of these peaks, but what we're striving for is consistency. So by correcting for each one of the inconsistencies that we see in those 3 samples, what we strive to get is the same result for every one of the 9 peaks I'm showing on the screen here, or every time we analyze this particular 2 compounds. Again, the goal here is to do consistent integration, thus making sure that any real changes that we measure in both the 1 and 2 forms are, again, high quality data and consistent measurement. Once the data has been collected, it goes through a series of both computational and manual QC steps to eliminate the noise, identify the metabolites present, ensure those identifications are real, and remove process artifacts. The most important step is one we call chemocentric data analysis. If you can imagine for a second, every sample that we analyze presents upward of 150,000 ion features from the mass spectrometer, represented by the circle on the left of the screen here. The first step in our process then is to try and identify those real metabolites from the soup or the noise. We run the data through an automated library that contains somewhere upwards of 4,000 named compounds. That then has the parameters set wide open, and we identify as many metabolites as possible. The goal here is not to screen things out, but to be as inclusive as we possibly can for every metabolite that we can identify. Once that's complete, there are 3 manual QC steps that the data goes through, leading to the identification of as many real metabolites in the sample as possible. In the example I'm showing here, we're identifying approximately 700 real metabolites. That data then goes into statistics. The important 50 metabolites, those that are being up or down regulated, are then brought out within the statistics, and 3 to 5 nodes are followed up on during the interpretation. As I mentioned, our library contains about 4,000 named compounds, most of which we have purchased as authentic standards, some of which we have synthesized in-house, and all of them have been run through our platform through all 4 methods. Anytime we change anything within our methods or within the technology that we run through every one of those 4,000 plus named compounds to re-identify them and ensure that any changes can be quickly mapped back into the library for future identifications. About 10 years ago, a group of metabolomics practitioners met and published a paper listing the various tiers that they agreed made up the identification of metabolites during an untargeted study. I've listed those 5 tiers here, everything from the top tier where you have an authentic standard run through the system down to the single unique features at tier 5, the lowest tier. Since we have purchased and analyzed the majority of compounds in our library, most of our metabolite identifications are tier 1 identifications, the best achievable, and we note within our library and in reports that we provide back to our clients when any tier 2 identifications are made. Once data has been passed through the automated library searching tool and the manual QC steps, it is then passed on to statistics and then back to the study managers. Those same study managers that were involved in helping with the study design at the front end of the process. We have about 20 PhD biochemists who work on the interpretation of data, mapping compounds that are up or down regulated to the actual biochemical processes and providing written reports, presentations, and publication support. So what you're seeing here on this screen on the left-hand side is not just an up or down heat map showing the regulation, up regulation or down regulation of compounds, but also the pathway maps that we have generated and provide back in the reports that we generate. That then leads to our total process, as you can see on this slide. So we've scanned through all of the metabolites within our library in each sample. We identify the markers that are up or down regulated, up being red, down being blue, and we focus on those markers that together provide an insight into what is going on in the body. In this case, you can see that we have measured many markers that are up or down regulated, scattered across the entire biochemical pathway map. These include markers of diet, external factors, and disease. Your eye is immediately drawn to the top left-hand corner, where a series of markers are both up and down regulated around a particular enzyme. In this case, branched-chain ketoacid dehydrogenase. These markers are a signature for maple syrup urine disease, an inborn error of metabolism. As I said, our library contains over 4,000 named authentic compounds, allowing us to assay for a huge variety of pathways, everything from lipid metabolism to microbiome compounds, and including common drugs and their metabolites and other external compounds. Since Metabolon has been in business since 2000, our first platform was actually put into operation in 2005, and as you can see from this slide, we're not very proud about the data that we generated. Back in 2005, when that first platform went online, we were only able to analyze for about 100 compounds, and our RSD across that platform was about 48%. So it was rather a guess as to whether or not we had correctly identified those compounds. Obviously, we've been diligently working since then to improve upon the quality of the results and the data that we generate through a huge series of technology, software, hardware, etc. Improvements that we've made. Today, with the combination of our Precision Metabolomics platform and the complex lipid platform, we measure between 2,000 and 2,500 discrete metabolites in human plasma, with an RSD of less than 5%. Now, each one of the measurements that you're seeing on the bottom left-hand corner there is from a pooled plasma sample. There's one of our QC samples that gets run as part of the 40 samples we run every day. We've been collecting pooled plasma samples and running that pool of plasma for many, many years now, tracking from one pooled set of plasma to the next. As I said, back in 2005, we saw about 100 compounds in that pooled plasma sample with a 48% RSD, and tracking through the years now, 2016, 2017, we're at about 4.5% RSD for over 1,000 molecules in human plasma just on the HD4 Precision Metabolomics platform, and then about 1,000, 1,100 lipids on our complex lipid platform. So to summarize this section of the presentation, our Precision Metabolomics platform produces the highest quality data with Tier 1 identifications through a combination of hardware, software, infrastructure, and rigorous process control and monitoring. This produces the highest quality data, and more importantly, it produces actionable biological insight. What I'd like to do now is move on to the second part of the presentation that focuses on how you can actually use that data. So really, we're taking precision metabolomics into precision medicine. Last year, Metabolon launched its first precision medicine product, Meta-IMD. This product uses the power of the Precision Metabolomics platform to analyze human patient samples for inborn errors of metabolism. Currently, we are able to scan for over 65 inherited disorders from a single 100 microliter plasma, urine, or cerebral spinal fluid sample. This has come about through 2 years of collaboration and development with Baylor Genetics at the Baylor College of Medicine. So I'd like to discuss first off exactly how we came to this point. 3 years ago, we were challenged by Art Burdette at the Baylor College of Medicine to analyze patient samples. Art had seen presentations on our processes and believed that we were capable of analyzing single patient samples. He challenged us to analyze 200 samples, blinded to us, 70 from unaffected patients, and 130 from diseased patients. As you can see, we were able to identify all 70 unaffected cases and 129 of 130 diseased patients. In addition, we were able to identify the diseases in each of those 129 patients. Now, the one missing patient was on therapy, so we feel a little bit cheated. In fact, what we were able to do is measure some of the treatment effects, but we weren't able to see the disease effects in that one treated patient. So here you can see a list of all of the diseases that were present and correctly identified. At the bottom, you'll see the one of the 2 patients with VLCAD disease that we were unable to identify because of the treatment. From here, we worked with Baylor to develop our inborn errors of metabolism testing, showing that we were able to identify diseases in plasma samples for the most part. This included the identification of some diseases that traditionally required cerebral spinal fluid to be drawn from infants, and we were able to identify the disease using only the plasma sample from those patients, from those infants. Fast forward a couple of years, and we're now able to measure over 65 inborn errors of metabolism using our Precision Metabolomics platform, screening for compounds in affected pathways rather than individual markers that might highlight a disease or a group of diseases. And this is important. What we're measuring here is not just a single marker for each disease, but the entire pathway that's impacted, showing proximal and distal markers to the traditional marker being up or down regulated in the total pathway, so showing the total pathway being impacted by the disease. So what I'm showing here is one of the pages from our typical Meta-IMD report. In this case, the patient is suffering from argynemia. And here, what you're seeing is a typical pathway report. Here again, we're showing the metabolites that are up or down regulated, leading to the diagnosis of argynemia. Like I said, it's important that what we're showing are markers in the total pathway that are being up or down regulated, rather than just the traditional arginine as a measure for this particular disease. So why is that? So if I switch to this next slide, what you see are 4 patients, all of whom have been diagnosed with argynemia. If you look across just the arginine result, what you see is that the arginine result looks about the same for every one of the 4 patients. However, one of these patients, patient 1, has presented to the ER multiple times in 3 months with acute effects of the disease. And it's not until you go to the proximal and distal markers in the pathway for this disease that you see why, with large changes for that one patient, markers being heavily up or down regulated, particularly for that one patient versus the other 3. It's clear to see by measuring the pathway that what we're actually seeing is that this particular patient has a more severe form of argynemia, And by measuring the pathway, we are able to actually distinguish that and distinguish this patient for more acute treatment compared to the other three. Changing direction a little bit, we've completed a number of GeneWide association studies with our partners at King's College London and Human Longevity. The most recent analysis was of nearly 2,000 twins at 3 time points, with samples being analyzed by both whole genome sequencing and via our Precision Metabolomics platform. And then we mapped the gene variants to the metabolite levels. If you take a look at this radial plot, there are a couple of things that come out from this. And this data was published by Long and myself and others in Nature Genetics in April of this year. First off, we see 644 metabolites that have been identified with very good heritability, a measure of the genetic influence on the changes of those metabolites versus environmental impact. The other thing that we've seen is that over the course of our work with King's College on twin studies, we've gone from 38 associations linking our metabolites to the genes and their function and being able to help identify some genes of unknown significance, from 38 to 145 to now 246 gene metabolite associations. And in particular, in this particular report, what we see is 90 novel findings. We're able then to look at individual patient samples who received some whole exome or whole genome sequencing, look at the metabolism, and be able to distinguish whether or not there's something important linking the metabolite information to the gene metabolite information. In this first patient sample, one of the most important things we noted was that the whole exome sequencing did not report as significant the aldolase B mutation. The computational analysis of whole exome sequencing data did not show this to be significant. Apparently, what happens in whole exome and whole genome sequencing is a computational priority is attached to the data to try and take the vast amounts of data and decide what is important and what is not. Our analysis of the metabolites of this particular patient showed something completely different. As you'll notice, the sorbitol level for this particular patient is one of the highest we've ever seen compared to normal, while the fructose is also highly upregulated. When asked this particular patient if they identified as fructose intolerant, first thing they said to us is, well, I really don't understand what that means. So we switched to showing pictures of fruits that are high in fructose, and the patient immediately self-identified as self-treating, if you like. They didn't eat any of those fruits because they knew they made them sick. The importance here was that the metabolite information was able to show that this actual aldolase B mutation is very important. It leads to fructose intolerance for this particular patient, and the patient went through subsequent dietary planning to avoid future complications from fructose intolerance. Second patient sample here that we're seeing, again, this patient was reported by whole exome sequencing as suffering from xanthinuria, a potentially fatal disease. However, what we see in the metabolite information here is something completely different. The hyperxanthine on the left is practically normal. Xanthine is slightly elevated, and the urate is high. In fact, what we would expect for a patient that was suffering from a mutation around xanthine dehydrogenase is completely the opposite. Hyperxanthine should be highly upregulated, and both xanthine and urate should be downregulated. What we're seeing here then is that in all likelihood, the patient has found a way of compensating, their body has found a way of compensating for this mutation, such that the actual mutation of xanthine dehydrogenase is not important at all for this particular patient. Again, highlighting the importance of the link between the metabolite information from the Precision Metabolomics platform and the whole exome or whole genome sequencing. Now, what I've shown in the last few slides highlights the ability of the Precision Metabolomics platform to provide actionable results on individual patient samples and a complement to both whole exome and whole genome sequencing. So my last example in this section highlights that there are also many cases where the precision and accuracy of the Precision Metabolomics platform provides high-quality data and meaningful results in traditional case versus control metabolomics studies. We've been working with Dr. Elizabeth Ryan and her team at Colorado State for many years. Here you can see some recent publications using data that Dr. Ryan has received from Metabolon. What Dr. Ryan and her team have been doing is carrying out studies in neonatal pigs, which have a very similar GI tract to infants. And her studies have focused on rice bran in these pigs to show that the bran stimulates microbiome metabolites that in total protect against bacterial diseases of the gut, principally Salmonella. Now, Dr. Ryan and her team are trying to now link this data to see if rice bran fed to children in third world countries can protect against these particular diseases and therefore protect against malnourishment and stunting of growth. And she's in fact gone on to show that feeding of rice bran to infants in Mali has indeed protected the children against bacterial infection, and in these cases has led to normal weight gain and height gain of these previously malnourished infants. So what's next for Precision Metabolomics? The final part of my presentation discusses what we are doing to enhance Precision Metabolomics efforts at Metabolon. I'd like to talk about molybdenum. Molybdenum is one of our CEO's favorite metals, something that he asks about regularly over the last year, and has led us to make a case for implementation of ICPMS for analysis of metals alongside our Precision Metabolomics platform. So in the top right-hand corner, I'm showing you a nice picture of some leafy green vegetables. Those are the principal source within the diet of molybdenum. If you remember back a few slides, I discussed the xanthine dehydrogenase pathway. Molybdenum is a catalyst for the formation of xanthine oxidase family of enzymes, one of which is the xanthine dehydrogenase. In addition, molybdenum is a catalyst in the formation of sulfite oxidase, an important enzyme in the cysteine metabolism pathway. Reduction in the amount of molybdenum in the diet leads to a shunt of this pathway towards cysteine sulfate, an irreversible reaction, decreasing the amount of free sulfate and glutathione in the body. Glutathione is obviously an important factor in preventing oxidative stress, for example in the liver, where it helps protect against liver disease. So it's obviously important that we maintain a usable amount of molybdenum in the body, and being able to measure that is another important step in our provision of precision metabolomics data. During 2007, we have worked to implement ICPMS for the analysis of metals into our precision metabolomics process. We now offer quantitative measurement of 23 metals, including molybdenum, which our CEO is very happy about, from a wide variety of matrices, including plasma, urine, feces, tissue samples, and cells. Again, with a standardized set of digestion protocols and a very rigorously controlled process. Our next challenge has been to be able to analyze from dried blood spots. This has been an interesting project that has gone on for the entirety of 2017. So far, we've been able to show that we can effectively extract metabolites from these cards and pick up markers of inborn errors of metabolism, for example, PKU. The sample prep and analytical processes are in place, and the remaining challenge for this matrix is to determine how much hematocrit how much the hematocrit of the samples impacts the measurement of particular metabolites, and what we can do to control for hematocrit. If you look at the slide here, what I'm showing on the right-hand side is the impact of hematocrit. You'll see some very different blood spots on the lower right-hand side. And particularly one of the things we're now working through is how does that impact the data that we generate? Does the card thickness itself have any important impact on the data that we generate? Is blood spotting technique something that we need to control for? So we're now doing a lot of research, having been able to show that we can actually analyze inborn errors of metabolism using blood spot cards. We're now looking to do our research on how we can control for the hematocrit and be able to analyze these samples as part of our typical process. This year, we've also added a targeted drug screen to our Precision Metabolomics platform. This gives a quantitative measure of 19 drugs and their metabolism alongside the standard result from the platform for all of the metabolites that we measure. The important part here is that no different sample is required. We can run this as a standard part of our Precision Metabolomics platform. We just produced quantitative results for the 19 drugs or their metabolites that we're showing on the screen here, and then the standard result for every other metabolite up to 1,000 metabolites plus that we see in human plasma. And the goal here is to be able to measure these 19 drugs and their metabolites for particular patients to see if any particular patient is suffering from an intolerance or a toxicity to any particular drug. Obviously, there's been a lot of reporting around the issues around particular statin use and whether people are able to take statins for the length of time they need to take statins. The same thing for Tylenol and other pain medicines. And so we've included those in our platform analysis, being able to measure particular patient samples and show whether the patient is normally metabolizing those drugs or not. Lastly, we've added analysis of sebum to our repertoire. So for those who don't know what sebum is, if you run your finger down the side of your nose, you end up with a waxy coating on your finger. That waxy coating is sebum. It's the lipid-rich coating on the surface of the skin. It's used as an antimicrobial, photoprotective, and wound-healing compound or set of compounds that lies on the surface of the skin, and it has a very unique lipid component, including some wax esters, sapienic acid, a very strange fatty acid, and squalene. Traditionally, sebum has been measured for skin care, for acne, and for cosmetics, but it's also being shown now to be a non-invasive readout for whole-body lipid metabolism. We've now designed processes to analyze sebutapes in both our Precision Metabolomics and our complex lipid panel. We take the same method and technology as our complex lipid panel. We've developed a targeted list that composes those wax esters, squalene, and free fatty acids that are important in the sebutapes. We analyze using a very standard extraction protocol, analyze the samples through our complex lipid panel looking for those particular classes and compounds. And then we've also added a fatty acid methyl ester panel using a GCMS as a targeted assay, and that is there principally for the analysis of those fatty acid components, particularly that sapienic acid that is very unique to the sebum. In the same way, we're able to take the sebutapes and place them onto the Precision Metabolomics platform. In the example I'm showing here, we've analyzed approximately 300 metabolites that were measured threefold over the blank sebutapes, and I'm highlighting here the xenobiotics, the drug compounds that we're able to see off of the surface of the skin in the analysis of 16 individuals with the sebutapes. Now, the important part here, as I said, is it's a non-invasive technology. You take the sebutapes, you wipe the forehead of the patient with a swab, apply the sebutapes for 30 minutes, take them off, stick them back onto the card that they came with, and then you can send them for analysis. So a very non-invasive technology for the analysis, in this case, like I said, of over 300 or so metabolites on the Precision Metabolomics platform, and then approximately 1,000 metabolites using the complex lipid panel. So, in conclusion, what we have developed here at Metabolon is a very high-throughput, industrialized, untargeted precision metabolomics process capable of precisely measuring somewhere between 1,000 and 2,500 compounds in human plasma with an approximate capacity through our headquarters of over 200,000 samples a year. We built everything into the process, very high rigorous processes, SOPs, quality control measures, infrastructure, hardware, and software to allow us to analyze single patient samples, which we started doing with the launch of our Meta IMD product. And we're under continuous development, including the addition of the ICPMS, sebum analysis, and dried blood spots to be able to assist further with the diagnosis and treatment of patients. I'd like to finish up by thanking some of my colleagues. In particular, I'd like to thank Elizabeth Ryan and her team at Colorado State for the data that she's allowed me to share with you. John Riles, Mike Milburn, CEO and Chief Science Officer, my research and development team headed by Annie Evans, project management groups headed by Rob Money and Kirk Beebe, the analytical team headed by John Lennon and Don Harvin, and then sales and marketing team, including Karen, who's on the line with me today. And I thank you for your attention. If you would please type any questions that you have into the question field, we'll be happy to answer them.
Thanks very much, Luke, for that really interesting presentation. As Luke mentioned, we do have some spare moments for some questions. So if anything sprung to your mind there that you want to ask Luke, please submit it into the question box at the bottom, and I'll be happy to relay that back to him. But one of the first questions we've got here is asking, can you also run tissue? What other types of samples are appropriate?
Yes, thank you very much for that question. It's a question that I get asked reasonably often. So Metabolon has been able to analyze over 350 different matrices to this point. We've analyzed everything from hair sample to toenail and pretty much everything in between. I don't think we've analyzed earwax, but we have analyzed nasal lavage, obviously plasma serum, fecal material, tissue samples, liver, kidney, skin samples, as I mentioned. We've also analyzed from a variety of different animals, including cat, mouse, rat, dog, monkey. We've also looked at a variety of consumer good products, PepsiCo products, orange juice, squirrel brain. We've looked at 2,000-year-old mummy samples, including the scrapings from the dental area of 2,000-year-old mummies. Dinosaur bones and rocks are a couple of the favorites that I've been asked about, although we didn't see much on rock. I think somebody was trying to recreate Jurassic Park, and we didn't see much in the dinosaur bones, so that didn't provide, I think, the results that they were hoping for. But yes, we've analyzed over 350 different matrices, pretty much everything that you can imagine.
Brilliant. And another question that's been asked you, Luke, is what sort of sample volumes are needed and how many technical replicates do you need to run?
Sample volume is very small. Typically, we're analyzing 100 microliters of plasma serum, urine, liquid samples. It's 100 microliter equivalent packed cell pellet. Obviously, that size is dependent on the size of the cells. It's a 100 microgram tissue sample, so it's those sorts of equivalent. Technical replicates actually is something that because of the QC processes and the quality control steps that we've taken, we don't need to run technical replicates. So we're actually running N of 1 sample. So there's no need to include into your study a series of technical replicates on each sample.
And many of the examples that you showed today were more around rare disease. What type of work have you done in more common disease?
Thanks, that's a great question. We actually have a big presentation, and I'm happy to share with other people on the numbers of different diseases that we have categorized. Internally to Metabolon, we have produced a diagnostic test for pre-diabetes. And now kidney disease. We've looked at NASH, we've looked at other liver diseases. Obviously, we've looked at skin diseases, including acne. So there's a wide variety of different diseases outside of the rare disease space that we have analyzed.
Okay, and there's a lot of interest at the moment in the microbiome and its role in disease. Do you detect microbiome compounds using your technology? Great
question, thank you, yes. So microbiome is obviously a very important part in the field right now, something that a lot of people are very interested in. As I presented on the work that Dr. Ryan and her team are doing, it's something that we've been working on for a number of years now. Typically, we see something of the order of 70 to 80 discrete microbiome compounds, those compounds that we know are not endogenous to the human, but are seen within human plasma samples when we analyze them. Within our library, we have something of the order of 240 or so discrete microbiome compounds. Again, compounds that are not endogenous to humans that we would analyze for when we analyze a human sample.
Okay. We've had a few questions be submitted just whilst you've been answering those ones, Luke. So I'll just start by saying how successful is the current metabolomics approaches in identifying toxicology aspects?
So we're somewhat successful in that space. We like to say that we can use metabolomics, particularly the untargeted approach, for pseudo-toxicology approaches. So we can, because we're obviously able to measure pathways, we are able to see changes within those pathways, particularly liver tox and the changes in the bile acids and kidney tox. We are able to assist in toxicology approaches. One of the things I have been very keen to do within Metabolon in the future is use a targeted approach. Obviously, we measure a lot of the bile acids, and we have a bile acid functional panel, which is a fully quantified targeted panel for analysis of bile acids. So one of the things I'm keen to do then is set up a set of toxicology type functional panels, so fully quantitative targeted assays for analysis of markers that we know to be important in both kidney tox and liver tox.
Mm-hmm. And another one that we've just had in, a question asks, what is required by the customer in terms of sample preparation, both for tissue and cells?
So the real key point for us when we work with all of our clients is the consistency of sample handling. We'll take tissue samples directly into a centrifuge tube or into any kind of tube that the client is willing to send us, just like that. We don't particularly require them to do anything more than flash freeze the tissue sample and then send it to us. The important part, as I mentioned really, is the consistency of the approach. If every one of the samples, let's take a plasma sample, for example. If you're collecting a set of plasma samples and the first set of plasma samples is going to sit at room temperature on the bench, then that's fine by us. What we would like to have happen is that every one of the samples stays at that same temperature on the bench until the samples are shipped to us. Now, if they're going to be frozen, they're frozen together, shipped to us on dry ice that way, and then we treat them from there. The real key to reducing the error that we see in the analysis is in the consistency of how the samples are handled before they reach us, as much as the processes and everything that we've put into place at Metabolon to maintain consistency throughout our process.
Mm-hmm. And another question asks that they notice that there's a lot of lipids that can be precisely measured. Do you use standards for measuring every one of those?
So we have two different processes, as I've mentioned, for measurement of lipids. We have the lipids that we measure within our Precision Metabolomics platform. Those are measured on one of our two positive electrospray methods. They are typically the sorts of lipids, heats, HODs, those sorts of things, cholesterol, things that are particularly measured in inflammatory situations. Then we have our complex lipid panel, which I haven't really spoken about today, that measures up to 1,100 complex lipids, triglycerides, cholesterol esters, as I mentioned in the sebum analysis, wax esters. For all of the lipids that we measure on the HD4 Precision Metabolomics platform, where we have called them a Tier 1 identification, we have a standard for them. For the lipids in our complex lipid panel, what we have done is utilized MRM technology to identify the lipids, so we don't necessarily have a standard for everyone. When we run the complex lipid panel, we also we produce a quantitative result, so we also have a set of internal standards, although we do not once again have an internal standard for every one of the lipids that we're measuring. Typically, we have a set of custom-designed internal standards for the complex lipid panel that includes somewhere around 8 to 10 internal standards that cover the breadth of sort of fatty acids connected to the head group in the lipid space. So I hope that answers the question. There are standards for a large number of the lipids measured on the Precision Metabolomics platform, but we're using MRM technology on the complex lipid panel to identify the lipids in that panel.
Brilliant. We have another question in here that asks whether you think NMR-based approaches have a place in the future of metabolomics and as well as in the application to precision medicine.
So I don't have an NMR on site. I have done NMR in my past at GlaxoSmithKline. Do I think that NMR has a place in metabolomics? Sure. NMR, my understanding of NMR metabolomics analysis is in fingerprinting, so measuring changes that happen rather than giving a full screen of every compound that is detected. And in that regard, I think it fulfills a useful role. So much so that one of the things I'm thinking about doing as an R&D project for this coming year is a fingerprinting technology where we simply take the sample and analyze it very quickly through direct injection into the mass spectrometer. And again, all we'll be measuring in that case is relative changes between case and control rather than giving a scan of every compound that we can detect. Those sorts of things are useful, I think, in triaging samples to determine which things you're particularly interested in, either for deciding to go down the road of doing a full untargeted metabolomic screen or for targeted assay work.
And we have one last question here that asks whether you have any system or program that can do disease predictions such as type 2 diabetes or any other good biomarkers.
Sure. So as I mentioned, we have a number of programs in-house that have taken our Precision Metabolomics platform data, developed fully targeted quantitative assays against those markers that we identify against a particular disease, and then move them on into a diagnostic test. So if I take the case of diabetes, for example, we identified through analysis of samples run against the euglycemic clamp approximately 23 markers that were up or down regulated against the clamp data and highly correlative to type 2 diabetes or pre-diabetic situations. We then took those 23 biomarkers, we designed a series of fully quantitative targeted assays against those 23 markers, ran again the set of targeted assays against the euglycemic clamp standards.
Okay. I found something on the web for of 20 quantitative time divorces against A20 primo.
Sorry about that. So we ran our targeted assays against the training cohort, correlated the data back to the euglycemic clamp data. We then ran a separate training set to, again, correlate all of the biomarkers and place them in rank order against the euglycemic clamp data. This resulted then in 3 biomarkers that were correlative to pre-diabetes and very early insulin resistance measurements of patients that might eventually progress through the pre-diabetic stages to full-blown type 2 diabetes. We've now developed a test within Metabolon that allows patient samples to be analyzed against those 3 biomarkers, and we give a score that shows how insulin resistant the patient is and shows early onset of pre-diabetes leading towards type 2 diabetes that allows the patient to be treated at a stage when the pre-diabetes can be reversed, as opposed to moving on to full-blown type 2 diabetes when it's most likely that the disease cannot be reversed.
Great. I'm afraid that is actually all we've got time for today. I'd like to give a big thank you to Luke for his time, and thank you to everyone who's tuned in to listen. We hope to see you all again very soon, and have a great day. But don't forget that you can tune in to listen to the webinar on demand any other time ahead of now. So take care.
Thank you.
I don't know what the hell that was.